dung beetles are a very important group of insects, and if we do not have dung beetles, the forests and floors will just be covered with poops of animals. So for this particular project, we wanted to test as to whether dung beetles can be used as sources of environmental DNA to detect the diversity of vertebrates within tropical rainforests, and also as to see if we can establish those direct interactions between individual dung beetles as well as the kinds of vertebrate and mammal dung that they are fed on. Firstly, we actually wanted to find what is the most optimal time frame to sample these dung beetles so that we can try and get as much DNA from their gut contents as possible. What we did was we actually conducted a feeding experiment. So we collected a bunch of dung beetles from the wild, and then we had to get the dung beetles to purge their guts so that their guts would be free of the DNA of any kinds of previous vertebrate dung that they had fed on. And then we force-fed them with elephant dung. So we had 8 different time points, and across these time points we randomly selected and then preserved these dung beetle species. So this feeding trial experiment had pretty interesting results. Like when we look at their guts visually under the microscope, the dung beetles that were collected within 3 hours post-feeding, we found that the amount of dung that is present in their guts was still considerably quite high. Like from the 6-hour time point onwards until the 24 hours, we saw that the proportion of dung beetles that have full guts kind of dropped pretty drastically. This kind of shows that their digestion time frame is kind of short, and so the most ideal sampling time frame has to be pretty short as well. However, a really interesting finding that we kind of found was that even dung beetles that were sampled 24 hours after feeding, there were a couple of individual dung beetles where we were still able to detect elephant DNA from their gut contents. So from this feeding trial, we managed to figure out what is the optimal sampling time frame to collect these dung beetles, as well as to optimize our protocols for sequencing DNA from the dung beetle gut contents. So now what we wanted to do is to test this approach in the wild as well to see exactly what kind of dung of vertebrate species that these wild dung beetles are feeding on. So to do so, we did a survey within the Central Catchment Nature Reserve in Singapore. We actually set up traps across 50 sites across this nature reserve. We set up traps both ground pitfall traps as well as canopy pitfall traps, because we are also interested to see if there was a difference between the communities of the ground foraging dung beetles as well as the canopy foraging dung beetles, and whether that will also influence the difference in the vertebrate species that detected from these 2 different dung beetle communities. So the traps that we used around these sites are actually live traps. So one of the main reasons why we use these live pitfall traps is because we really wanted to preserve the DNA of these dung beetles and the DNA of their gut contents as best as possible. So after 24 hours, we collected these live dung beetles and immediately we preserved them in like 100% molecular grade ethanol to preserve their DNA. So now that we're in the lab, it's a whole other process of processing these samples. So after dissecting out the gut contents of every individual dung beetle and extracting the DNA from those gut contents, the next step is to amplify whatever kinds of vertebrate DNA that can be found in those samples. So to amplify specifically vertebrate DNA, what we used were primers. So primers are also short DNA fragments that bind to a targeted region of the DNA that we are interested in. So in this case, we use 2 different types of primers. The first primer is also known as the 16S rRNA primer. That primer is more targeted to mammal DNA. And then we use another set of primers, also known as the 12S rRNA primer. And those primers are targeted to vertebrate DNA. From this particular field survey, we were successful in sequencing different kinds of vertebrate DNA from the individual dung beetles that we collected. From over 500 individual dung beetles, we actually were able to detect 18 different species or native vertebrates that are found in Singapore. So this was actually really interesting because we managed to detect the DNA of rare and elusive mammals. This included the endangered Sunda slow loris as well as the Sunda pangolin. That information, we were then able to construct the ecological networks that show these direct interactions between the dung beetle species and the vertebrate species in each of those 50 sites that we did our surveys across the Central Catchment Nature Reserve. And when we looked at these networks in more detail, we then found that these networks were actually pretty similar all across the Central Catchment Nature Reserve. So there wasn't really any difference in the structure of these networks across that space. But having said that, the fact that we were able to construct so many networks across this wide spatial area, the fact that we're able to demonstrate this really shows that dung beetles can indeed be used as an effective resource of environmental DNA, and then we can use that to construct these ecological networks. We managed to use this novel technology of DNA metabarcoding to detect vertebrate DNA from dung beetles as an environmental DNA resource. So this can be used in complement with traditional or conventional survey methods. So by using dung beetles as an indicator, it's also much more time saving because we just need to set up traps for a short period of time and then go through that whole molecular process. And if you're able to properly optimize those molecular processes, you can get your data in a relatively quick time frame. So therefore, having this method and using this in complement with those kind of traditional methods, it's like an added bonus to making these sorts of comprehensive biodiversity assessments.